Okay, now we're going to continue with our discussion of chemistry in Chapter 2. The topic is now organic molecules. Remember, we previously defined inorganic molecules as those that do not contain carbon and hydrogen. Organic molecules do contain carbon and hydrogen. Our first example of an organic molecule is carbohydrates. Carbohydrates, some examples are sugars, starches, cellulose, things like glucose, lactose. Those are all examples of carbohydrates. They do contain carbon, hydrogen, and oxygen in a ratio of 1 to 2 to 1. In other words, they have twice as many hydrogens as they do carbons and oxygen. Look at the name, carbohydrate. You can almost see that word carbon in there and hydrate referring to the H2O that's within it. It's literally an atom of carbon and a molecule of water joined together. They come in three varieties: monosaccharides, disaccharide, and polysaccharide. That suffix saccharide is Latin for sugar, so mono means one sugar. Disaccharide would be two sugars joined together, and a polysaccharide would be more than two sugars joined together. Here's some examples of these carbohydrates from your textbook. Dehydration, synthesis, and hydrolysis we mentioned before. Those are chemical reactions that involve water. Check them out here. We have the process of dehydration synthesis where we're taking this H2O group off right here. We're looking at a disaccharide molecule, a molecule made of glucose and fructose. Removing this H2O group right here that joins the molecules together by an oxygen, and now we have sucrose. So that's the process of dehydration synthesis, taking H2O out of molecules so that they can combine to become a larger molecule. Here's the molecule of water that was removed. In the opposite reaction, we're going to take this water molecule, bump it back in there on that little oxygen that binds them together, and break those two molecules apart to create a molecule of glucose and a molecule of fructose. That, remember, is called hydrolysis. It's the technical term for digestion. That's when we're breaking molecules down. That suffix lysis literally means to break. In the second diagram here, you can see lactose. Lactose is a disaccharide molecule made of a molecule of galactose and a molecule of glucose. Then we have maltose here. Maltose is another disaccharide made of two glucose molecules joined together. Check out glycogen here. Glycogen is actually a heavily branched chain of glucose molecules as opposed to cellulose, which is just a straight chain of glucose molecules joined together. Glycogen and cellulose would be examples of polysaccharides. As I mentioned, monosaccharides literally translates to one sugar. Some examples are glucose, galactose, fructose, ribose, and deoxyribose, which are important sugars in DNA and RNA. Glucose, of course, is our main source of energy. That's the energy that we get, or the molecule that we get when we break down the majority of our food. And that's going to yield some ATP, which is the usable energy in our bodies. Disaccharides are made of two sugars together, like glucose and galactose, make the sugar lactose, which is milk sugar. Those molecules of lactose are broken down by an enzyme called lactase, and that enzyme will yield a molecule of glucose and a molecule of galactose. That's, of course, through a hydrolysis or digestion reaction. Polysaccharides are large, complex carbohydrates, so they're made of many sugars. From tens to 100s of monosaccharides can join together. Example in humans is glycogen. When your body's bringing in lots and lots of carbohydrate molecules and it doesn't know what to do with them because you're not exercising or you're not using all of that glucose, it's going to store it in a form called glycogen. So that glycogen is going to be stored in your liver as well as in your skeletal muscles, which makes sense because if you are exercising or you are doing something that requires immediate energy, you want that glycogen to be readily available to your muscle cells so that it can be broken down into glucose and then translated into ATP to be used as fuel and energy for the body. Starch and cellulose are the complex carbohydrates within plants. Cellulose is a part of plant cell walls, and these create the loose term fiber that we use in our diet. When we're eating plants, and that roughage that we're eating is that cellulose, and we tend to just call it fiber in layman's terms. Lipids are another type of macromolecule or large molecule that make up our cells. These are molecules made of carbon, hydrogen, and just a little bit of oxygen in there. Since there's not a lot of oxygen, the majority of these molecules are actually hydrophobic, which is why they are hydrophobic molecules and they do not dissolve in water. They will actually dissolve in fat. They are fat. Oils will dissolve in other oils. Things like triglycerides, fatty acids, phospholipids, steroids are actually a fat molecule. Other fat-soluble vitamins like vitamins A, D, E, and K are also a lipid derivative. Triglycerides are molecules made of glycerol and 3 fatty acid molecules. They're literally formed by dehydration synthesis reactions, those molecules or reactions rather that take water away from molecules to join them together. The fatty acid chains can be saturated or unsaturated. A saturated fatty acid will have single bonds throughout the carbon and hydrogen interactions, whereas unsaturated fatty acids will have at least one double bond. They could be monounsaturated, where there's just one double bond, and polyunsaturated, which would be many double bonds. The big difference between saturated and unsaturated, first of all, most of you probably know saturated fatty acids are not as healthy for you, whereas the unsaturated fatty acids are better for you, if not even good for you. Things like olive oil, fish oil, those are all very nutritious forms of fat. They're liquids at room temperature, as opposed to saturated fats, which are things like butter and lard, animal fat. Those are going to be solids at room temperature, and that has to do with their chemical properties. Here you can see what I was talking about with the structure of a triglyceride. You see the glycerol molecule here, and then the fatty acid tails, which are mostly carbons and hydrogens. This one has a little kink in it or a bend. It's a different type of monounsaturated fat because there's one double bond within that chain. Phospholipids are the special type of lipid that make up cell membranes. Every single one of our cells is surrounded by a double layer of phospholipids. There's nonpolar fatty acid tails, and then there's a polar phosphate group, which make up the heads of these structures. I have a little diagram here for you. Here you can see what I was mentioning with the head, the polar head, and remember it's polar because it has a mix of various size atoms in it. We got some nitrogen, some carbon, some hydrogen, some oxygen, some phosphate in there, which make it polar, whereas these tails are mostly carbons and hydrogens, which are going to be the non-polar area of the molecule. Check out this one, which is a monounsaturated fat there with that double bond. These phospholipids are going to basically form a sandwich double layer membrane surrounding the cell. Notice that they're pointing in opposite directions to again create this kind of sandwich effect where we have the polar heads on the outside and the non-polar or hydrophobic tails in that inner part, or the peanut butter and jelly part of that sandwich. Steroids, as I mentioned, are another type of lipid. Cholesterol is a type of steroid. Most students don't realize that, but it is. It's important in the cell membrane structure. It's an important building block for many of our hormones as well. Testosterone, estrogen, as well as vitamin D, all use cholesterol, which is a steroid in their synthesis. Here's an image for you just to show you what it looks like. Again, what we're looking at here is all of the carbon and hydrogen together, which make it a lipid. Proteins are another group of macromolecules which are important in our body. Basically, everything that's in your body. How you describe yourself is basically based upon the types of proteins that you have. Your hair color, your eye color, your skin color. You have all of that because you have those particular proteins. So if you have curly hair, it's because you have this type of protein. If you have straight hair, it's because you have that type of protein. Proteins are really what make us, us. They're made of carbon, hydrogen, oxygen, nitrogen. Sometimes there's sulfur in there as well. They can be fibrous proteins, like the type of protein that makes up our hair. They can be globular proteins, like the type of proteins that work as enzymes in our body. They also serve specific functions, as I mentioned. They can serve as certain parts of our cells. They act as antibodies, helping to fight in our immune system. They act as enzymes to help with digestion, hormones as well, to help send signals and control the body, and they're also important in muscle contraction. So let's look at some different types of proteins. First, let's look at the structure of proteins. Proteins are made of amino acids. Amino acids are the building blocks of proteins. They're formed by special bonds called peptide bonds. Here you see one amino acid and another amino acid being formed together by dehydration synthesis, forming a dipeptide or 2 amino acid molecule. And here's that water that was extracted in that reaction. Our amino acids come together in something called a primary structure. A primary structure is literally just the sequence of amino acids within a protein. So again, many amino acids are going to come together to create a protein. Some proteins can be hundreds of amino acids long. The literal sequence of amino acids or the types of amino acids that are joined together is what makes up the primary structure. There's 20 different amino acids in our body. So they're kind of like letters in a word or words in a sentence, combining in different ways to make different types of proteins. The secondary structure is when those amino acids kind of twist and fold onto one another through hydrogen bonding. The tertiary structure is now when disulfide bonds and other hydrogen bonds give our amino acid sequence a more three-dimensional shape. The quaternary structure would be when other molecules come into play and interact with this tertiary structure. Things like iron might come in to our protein called hemoglobin to finalize it. Hemoglobin, of course, is our protein that carries oxygen in our blood, and it's because of that iron that binds to it. Let's check out what I was talking about with these structures here. Here you see a number of amino acids coming together to make up our primary structure. Each one of these amino acids is different. Again, remember I said there's 20 different amino acids that can arrange themselves in different ways in different quantities to create all of the thousands of proteins in our body. Then these amino acids are going to hydrogen bond to themselves, to each other, and form, sometimes they form a helical structure like this. It's called an alpha helix, where they kind of twist around each other. Almost looks like DNA, but it's not. This would be the secondary structure. The tertiary structure here is when we get some more folding and bending and twisting of our molecule. And then the quaternary structure is when we maybe have other polypeptide chains come together or other molecules or atoms come into play. As I mentioned, iron coming into the hemoglobin protein to finalize that. Again, the sequence of amino acids within a protein is ultimately what's going to determine that protein's function. If we just flip back here for a minute and take a look at this image where we have all of these hydrogen bonds creating this helical structure, well, if one of these amino acids was slightly different, that alpha helix might not form in that same pattern. We might get more of this beta sheet or a pleated pattern as opposed to a helical structure if one of those amino acids was different. So the specific sequence of amino acids is imperative in the overall structure of a protein. For example, sickle cell anemia, a disease in which some of you may be familiar with, is really just due to one single amino acid substitution in the hemoglobin protein. In other words, of the 120 or so amino acids that make up the hemoglobin protein, just one of them is incorrect to give you sickle cell anemia. Ultimately, the shape of that protein is held together by those hydrogen bonds, which are going to determine its function. Things like temperature, pH, radiation, etc. can cause a process called denaturation, which is basically when those hydrogen bonds are breaking. Think about our hair, for example. Ladies, you probably know what I'm talking about, right? Think about those humid, hot days. Our hair is not always acting the way it should be in those days, and that's because the temperature and humidity is affecting the actual amino acids in the protein of our hair, making those hydrogen bonds form in different ways, making our hair a little more unruly than it normally would be. So because of things like temperature, pH, etc., those amino acids can change and ultimately change the function of the protein. Something like a cooked egg is another good example of a denatured protein. The reason eggs cook and turn from that clear color to that white color is because the protein, which is called albumin, is actually being converted and denatured in that cooking process. The process of heating that egg white is actually changing the conformation of that protein. Proteins also act as enzymes in the body. Enzymes are catalysts. They're there to help reactions take place. Ultimately, if you just swallowed a cracker and you didn't have any digestive enzymes, over time, 10 years or so, those cracker molecules would eventually break down. Unfortunately, your body has high energy demands and requires that that cracker or sandwich or whatever molecules you brought into your body be broken down very quickly because you have lots of things that you need to do that require energy. Luckily for your body, enzymes are there to speed up those chemical reactions, so things like digesting a cracker take merely hours instead of years. Enzymes are typically named after the molecule that they work on, so enzymes are very specific. They're only going to act on specific enzymes. You can't take the enzyme that's used to digest milk protein, for example, it's called lactase. You can't take that enzyme and expect it to break down maltose or expect it to break down a galactose, another disaccharide molecule. It won't be able to do it. Enzymes and their substrates fit together like a lock and a key. They're very, very specific. Enzymes also require specific temperatures and specific pHs to work within. Just like I was mentioning on the last slide with temperature and pH affecting the denaturation of proteins, since enzymes are proteins, if the temperature changes or if pH changes, those enzymes aren't going to work as appropriately as they were before. That makes sense if you think about anytime that you're sick or you have a fever in your body, what's going to happen to your digestive enzymes? They're not working right, which is why sometimes when you have a fever, your stomach is also upset and hurts, or you're not digesting things as well because those enzymes aren't working. Enzymes usually end in the suffix -ase. Lactase is the enzyme that breaks down lactose, for example. Enzymes, I'm looking at this slide. I'm pretty sure I described all of this already. Enzymes bind to their substrates at a specific area called an active site. When the enzyme fits into the substrate, or the substrate fits into the enzyme, I should say, something called the enzyme-substrate complex is formed. Literally what that is, if you can imagine the lock and the key, that's when the lock and key come together. It's called the enzyme-substrate complex. Enzymes are going to catalyze those reactions to form bonds and break bonds. They can work in both directions. So in other words, they can create products, or they can put those products back together to form the reactants that they originally started with. The product is formed, that enzyme can be reused and recycled. That's the cool thing about enzymes, is they're catalysts only. They're not consumed in the reaction. One enzyme can perform the same reaction over and over and over again. Here you see what I was talking about. We start with our enzyme here, sucrase. Here's my mouse. Here's our enzyme sucrase. The substrate, sucrose and water, come into the active site. This is a process of dehydration synthesis because we're about to break apart, or rather hydrolysis, because we're about to break apart this disaccharide molecule. Here we have that enzyme-substrate complex. That enzyme's going to do its deed. It's going to do all that magic work and break apart the bond. And now we get products of glucose and fructose. And now our enzyme can be reused again, meet up with another sucrose and water molecule, break it apart, and then be used over and over and over again. Here's another animation of enzyme functions for you to view it on your own time. The last major molecule that makes up all cells is called nucleic acids. Nucleic acids are organic molecules that have carbon, hydrogen, oxygen, and phosphorus. They're made of smaller molecules called nucleotides. Each nucleotide is composed of a 5-carbon sugar, either deoxyribose or ribose, a phosphate group, and a nitrogenous base. There are 4 different nitrogenous bases that could be used. Actually, there's 5. That is actually a typo. There's 5 different nitrogenous bases that make up the different nucleotides. Let's talk about the 2 main types of nucleic acids first. Deoxyribonucleic acid, DNA, right? The NA in DNA stands for nucleic acid. Deoxyribo is referring to the fact that there's no oxygen, deoxy, on the ribose, which is the sugar that's part of this nucleic acid. DNA, of course, is the genetic material encoding instructions for protein synthesis. In other words, all of the proteins that make you, your hair color, eye color, skin color, height, right, the shape of your nose, the shape of your toes, etc. That all comes from instructions in your DNA. It's a double helix molecule which is self-replicating. In other words, it can replicate itself before the cell divides so that each new cell has its own copy of DNA inside of it. Ribonucleic acid, or RNA, is kind of like a little messenger or helper for the larger DNA molecule. We'll look at the functions of these more specifically later. Here you can see some of the specific structures that I was mentioning in reference to DNA. DNA is a double helix. You see that here. It winds around itself over and over and over and over and over and over again, condensing and condensing and condensing until you get these very condensed structures called chromosomes. Imagine that you're holding a string in your hand, and one end of your string is in your right hand and another end of the string is in your left hand, and you were to twist that string over and over and over again. What's going to happen to that string over time? It's going to twist and turn on itself and eventually get shorter and more condensed. And that's exactly what your DNA does to squeeze inside the nucleus of every cell. Some of the major differences between DNA and RNA, remember I said that there's actually 5 different nitrogen bases, not 4. There's adenine, cytosine, guanine, and thymine, as well as uracil. RNA has uracil instead of thymine, so that's one big difference between DNA and RNA. The sugar that makes up our, let me just plug my computer in, sorry my battery is going to die on me. There we go. The sugar that makes up these nucleotides is deoxyribose in DNA, hence the term deoxyribonucleic acid, as opposed to ribonucleic acid, which has the ribose sugar. DNA is a double-stranded molecule. It's a double helix, 2 sides of a twisted ladder, whereas RNA is not a twisted ladder. It's literally just half of a ladder, and it's a very short ladder as opposed to DNA, which is very, very, very long. In terms of base pairing, DNA, in the DNA molecule, A is going to pair with T and C is going to pair with G. In RNA, it's almost a misnomer to say that A pairs with U and G pairs with C, because there's no pairing in RNA. RNA is a single-stranded molecule, so there's no opposite side to the RNA molecule. We'll talk about that more later. DNA is self-replicating, as we mentioned. There's special enzymes that will help DNA make a copy of itself, and of course it's used for making proteins, whereas RNA is made by using DNA as a blueprint. And the main job of RNA is to carry genetic information out to the ribosome where proteins can be made. There's really only 2 types of DNA. There's nuclear DNA, that's the DNA that we're used to thinking about, the double-stranded DNA arranged in chromosomes inside the nucleus of every cell. But our mitochondria, which is an organelle inside of our cells, also has DNA inside of it. It's kind of a different structure. It's circular, doesn't have as many genes on it, but that's there as well. Our RNA comes in 3 different varieties. There's messenger RNA, always written lowercase m, capital RNA, transfer RNA, or tRNA, and ribosomal RNA, which is rRNA. We'll talk more about those in Chapter 3. Last important molecule to talk about in this chapter is ATP, Adenosine Triphosphate. As you can imagine, it's made of a molecule of adenine, which is the same nitrogen base that we saw in the DNA molecule, a sugar, ribose, just like in our RNA molecule, and there's 3 phosphate groups, hence the name triphosphate. Why is this molecule important? This is our energy currency. Any cell that wants to talk energy, any cell that wants to do something is going to do it using ATP as its energy currency. So, how does this happen? ATP releases energy to become adenosine diphosphate, ADP. Then, when the cell is absorbing energy, in other words, you eat something and break down glucose to create more ATP, that energy is going to be captured in ADP and create ATP again. Let's see if I can get fancy here and draw this out for you. I haven't done this before, but we'll see how I do. So we have ATP, ooh, sorry, this is really bad. ATP creates ADP, right? ATP is adenosine triphosphate. ADP is adenosine diphosphate. So there's only 2 phosphate molecules. The third phosphate will draw right there. ATP makes ADP plus P. And this is a cyclical process. So in other words, energy is released. Energy. Ooh, this is really bad. Sorry, guys. This says energy. I'm using a touchpad mouse, so bear with me. Released. Oof. Very sad drawing. Energy is released. And over here, energy is captured. Getting a little better. Energy captured. A very sad drawing. So let's talk about what this actually means. In other words, ATP, this is our high energy molecule, is going to be used for muscle contraction. So let's talk about an eye blinking, right? When you blink your eyes, your muscles are contracting. Boom. This happens about 20,000 times. About 20,000 ATP molecules are broken down every time you blink one eye. So really that would be a wink, right? So you're releasing energy in that eye blink to create ADP plus P. Then your cell breaks down some more of that glucose that is waiting around, capturing some energy to create more ATP, and then boom, you can blink again. And then so on and so forth. This is a cyclical process that happens over thousands of times every minute, I would say, within the cell. The cell's using this ATP to do everything from making new DNA to dividing through mitosis, repairing itself, fighting off pathogens, pumping in glucose, pumping out waste products. This process is very important to the cell. Alright. And here's that same reaction, but I like my little diagram better, even though the font was hard to write for me. Here we have ATP, our adenosine triphosphate molecule, and water, creating ADP and energy. That last phosphate is kind of hanging out right here. ADP and P and energy to do whatever that cell needed to do. And this is a diagram showing you exactly as I was talking about, that energy is released, or this muscle is relaxed rather, and our cell has ATP, and then that contraction happens, and now we have ADP going on inside the cell. Here's another animation of the whole process at work for you to check out. And that concludes Chapter 2. See you next time.